Okay, here is the second lab for the fungi kingdom. This is where we're going to look at molds. One of them that you're going to look at could possibly be a yeast, but the rest of them are all going to be molds. So remember that the molds are going to be multicellular. They're going to have filaments. They're going to have spores, okay, as opposed to the yeast that were single-celled and did not have filaments. Like I said, the one that crosses kind of between the two that have pseudohyphae is your Candida albicans. So for this lab, each student will be given one tube of mold or yeast. I might have it just mold, yeah, I think I'm going to do that way. I think we're just going to only do molds. And so each student gets their own tube. They will not be labeled with the name of the mold. They will be just numbered, okay? So when you take your tube, basically you're going to write your number on your plate rather than the name. So one tube, one plate per student, okay? It's going to be up to the student to properly identify the mold. And you'll use your lab manual and the characteristics in the lab manual, and also what we talked about in lecture, to figure out which mold it is. Now, the lab manual has several different possibilities. However, there's some that we do not have. On the day of the lab that we do it, I will tell you the specific species, but I'm imagining it wouldn't be more than 5, okay? So there's going to be 5 possibilities, roughly, when we do the lab. So they'll be numbered. Most likely the lab person at your table will not have one of the same ones. I want each student to be able to identify them themselves, which means that when you go to identify them, you need to be able to tell what kind of spores they have, what kind of hyphae they have, whether they're conidiospores, whether they're sporangiospores. So you're going to be looking at those characteristics, whether they're septate or not, the different things that we talked about in class. Plus your lab manual has a key to follow to figure out which ones they are. There's the color of the colony and different things like that that you're going to use at characteristics. We are pretty far in the semester. By now, students should be able to identify things on their own. You're going to be left on your own, meaning that I'm not going to help you in identifying it. If you tell me what you have, I can say yes or no, you're correct, but I'm not going to help you in the identification process. So what you're going to do on the first day of the lab, it's very, very simple. First day of the lab, using your sterile technique, you're going to be just taking the fungus, the mold is going to be suspended in a Sabdex agar or broth. You're basically just going to go with your loop that's sterile, and you're going to go down and kind of, you may have to push it down a little bit because it might be kind of hard at the top, but basically the idea is just to get some spores. And then you're just going to basically open your plate and you're just going to touch the middle of the plate. You're not going to spread, you're not going to dig down, you're just going to touch. You're going to sterilize your loop like you would always do. Then for the plates, you're actually going to put them in the incubator right side up. And just like we did with the yeast, these are going to sit in the incubator for a week. Now some of these molds have a tendency to spread really, really, really fast. So what we'll do is we'll take a look at the incubator on Monday or something, and if they're growing too fast, we'll take them out, okay? Because in the next class period, you're going to have to look at this plate and you'll measure how far the colony grew. You're going to use the cultural characteristics that we learned in the very first lab, or I guess the second lab that we did in class. And you're going to look at the margin, is it undulate and things like that. What does the colony look like? You're going to look at color. You're going to look at the top side. You're going to look at the bottom side. And then when in the lab manual, when it says to use a hand lens, we're going to use the dissecting scope. And I mentioned in the yeast lab that I'd show you what the dissecting scope is. So for this particular microscope, it's very, very simple. You would plug it in like you always would. There are 2 light switches. There's the main switch, and then there's these 2 switches over here. The main switch just turns it all on, but these 2 switches, one turns the light off on the bottom, and one turns the light on, turns the light on at the bottom, and one turns this light on. So sometimes when you're looking and you have the light coming from the bottom, it's better. Other times it's better to turn off the bottom light and turn this light on. So you'll have to kind of play with that. This does not have the two different sets of lenses like we see with a compound microscope. It basically has the ocular lens. And then these knobs right here is kind of like coarse adjustment, but it will make it closer and further away, okay? Let me rephrase that. It'll make it look bigger and smaller. This is the coarse adjustment here. And so you basically can see as I move this, it's moving the entire body tube. And so you're to get closer or focus in and out. One of the things to be aware of is when it does grow on the plate, you might get a lot of condensation. So you may have to look around the edges or try to work around the condensation to see it. I would not recommend opening the plate and leaving it open because these do have spores and you don't want to leave it open for a long period of time. So to get your colony characteristics, you're going to look at it, you're going to put it under here and look at it, and that's how you're going to answer that part. The next part is going to be the actual microscopic view with your compound microscope. So it's the same as we did with the yeast. You're going to use the lactophenol cotton blue, and you're going to make a wet mount of the mold. Now what you want to be careful with is try to take it as gently as possible off the top. However, some of these molds, when those hyphae get digging into the agar, it gets really hard to pull them off. So in some cases you almost have to take a chunk of it to get it to work. In that case, you're going to try to spread it out as much as you can in the stain before you put the cover slip on. Then that is what you're going to look at with your compound microscope to get your microscopic view. Okay, so you're going to be looking at the view on the plate, and then you're going to be looking at the view at the microscope. By the end of the lab, you should be able to identify to the genus level which one that you had for your particular unknown.